This video covers Chapter 3, How Cancer Spread, and we are just talking about the last third of the chapter. So in this figure, we are looking at a mouse model where cancer cells were injected into the tail of a mouse. So that's shown here with number 1, inject melanoma cells. So these melanoma cells were collected from a source and they were put into suspension and then injected into the tail vein here. And so what we see is that those cells will travel through the bloodstream and settle somewhere within the mouse's body. And if they have the ability, then they will form metastasis. So in this first mouse, we see that there were 2 mets formed in the lung. And in this experiment, what they're doing is they are collecting the mets. Then they are resuspending them, injecting them into a different mouse. And in this case, what they see is that they actually form a little bit more mets than in the first case. We see 2 dots here, and we see 3 here. And then down here, we have the process repeated again. So our met was collected, it was resuspended, put back into the tail vein of a mouse, and then the metastases were analyzed. And we see even more. So we have 1, 2, 3, 4, 5, 6. 6 mets in this mouse. And we repeat that cycle, removing the lung mets and then injecting them into the new mouse until 10 cycles are complete. And at the end of that, what you find is that you have many melanoma cells that can form many metastases in the lung. So what is this telling us? We had started with a suspension of cells in step 1, and they could form a few metastases. And then we took those metastases, resuspended them, put them in a new mouse, collected them, resuspended them, put them in a new mouse, and did that over and over again. So we were basically selecting for cells that can form metastasis. And so this gives rise to the idea that perhaps in our first step up here, we had a collection of cells where we have many different types of cells available. They're all the melanoma cells, but maybe only one of these cells had the ability to form the metastasis. Maybe the other cells were lost somewhere in the process. Maybe along the way as they were traveling through the body, they didn't make it. And so that's kind of the idea, is that when you have a tumor, that there's going to be many different types of cells within that tumor, what we call clonal populations. And then only some of them have the ability to do metastasis. Only some of them have the ability to survive in the bloodstream, leave the blood vessel, invade into the new tissue, and then grow and divide in the new tissue. So this is kind of the idea that when we're looking at this experiment on the right, that we are selecting out a cell population, a clonal population, that is selected for the ability to undergo metastasis. And so if you select for these cells, then you will have a new population of cells that's full of cells able to do metastasis. And so that is called the clonal theory, which I have a diagram here below. So it's saying like, okay, at some point in time, we had all normal cells here, right? So these are all normal cells before cancer has arisen. And then there was a founder cell that evolved, so underwent mutations, and then had the ability to undergo unlimited cell growth. So over time, that's going to form our tumor. And then some of these cells will start to mutate and form kind of like a different clone where it has multiple mutations. So we're kind of seeing that as indicated by the red plus the purple dot here. It has like a couple different changes. And then you give it time, and you don't necessarily lose that first population, but you're gaining a second population here. And you give it more time, and then you have yet another alteration where you have kind of like another clonal population popping up. So we have all of these little different mutations that are occurring, but not every single cell in the tumor will have all of the mutations present in that population. You're going to have these subclonal populations, and some of them will have the ability to be more metastatic than others. We had mentioned previously in class that once a cell becomes metastatic and has the ability to enter into the bloodstream, that it will have the ability to travel through the body. And some of these tumors kind of have a predictable place that they will set up their metastasis. And this has to do with how they travel through the bloodstream or how they travel through the lymphatic system. So once a cell invades into one of these vessels, then obviously these two systems, the circulatory system, the lymphatic system, they each have kind of a path and a direction that fluid is moving in. And there is an intersection between the two. So even if one of these starts off in the circulatory system and travels through the arteries, out to the capillaries, into the tissue, but then it could even be reabsorbed into the lymph nodes, the lymphatic system, and then it can move up into the lymph nodes. So there's the interconnection between those two different systems. So even if it invades into one of them, it has the ability to spread through both of them. And sometimes if it's going through the lymphatic system, then sometimes it will kind of locate into these lymph nodes, which are located along the lymphatic system. You see all these different little dots here, and a lot of times those cells will kind of get caught up in the lymph nodes and kind of take root there. So like we mentioned before, cancer cells will metastasize to certain regions, not all the time, but there tends to be some patterns. And this is due to how the bloodstream carries cells and what direction through the body, the flow of the direction of the blood. If you follow the arrows here, you can see the blood is going from the heart. It takes a stop over to the liver. Then we have a stop at the GI tract, and just moving through the body. So there's a flow and a direction for how this blood is moving through the body. So it is possible that if you have a cancer in a specific area of body, that they can be trapped just in the first capillary bed that they encounter. So sometimes that is the case. And sometimes there are organ-specific conditions that can determine the site for the metastasis. So for example, bones can secrete factors that promote proliferation, and certain types of cells might really respond to that and be able to kickstart the process to have increased proliferation and tumor formation. As you can see down below, we have certain cancers that have a common metastatic site. So we have stomach cancer, colon cancer that's going to the liver. We have prostate and breast that tends to go to the bone, kidney and thyroid that tends to go to the lung, and liver that kind of has a broader range because it's going to be spread throughout the body. Now, it has been noted in many cases that metastases don't always occur in the same direction as the blood flow. So that a metastasis for certain cancers sets up in areas that maybe you wouldn't logically think is their first stop in their travel through the bloodstream. And so here we have the Paget seed and soil hypothesis. So this was established by Dr. Paget in the 1889, 1890s, where he stated that metastases do not occur randomly, but they occur via the seed and soil hypothesis he came up with. And this has to do not with the circulation of the bloodstream. It has to do with the fact that the cells are going to be traveling through the body. They will have access to different tissues in different areas, but they do not take root in all of those tissues and all of those areas. And certain cancer cells tend to take root in certain places. So thinking about that, it's kind of like thinking about having a seed. Like if you had plants and you had plant seeds, and then you were going to spread them along the ground. If you spread them into like rich, nutrient-rich, moist soil, then they have a greater chance of growing. If you drop them into sand or in soil that's nutrient depleted and not moist, then they're not going to have a chance of sprouting, right? So it's the same idea with these cancer cells. If you put them into a tissue that is not secreting growth factors and the type of things that cancer cell requires for it to grow, then that cancer cell is not going to survive. But if the cancer cell lands in a place that is excreting the right factors, the right growth factors, the right cytokines, the right hormones, then that cancer cell can survive, it can proliferate, and it can really just take off. So that's the seed and soil hypothesis, right? So the seeds would be the stem cells or progenitor cells. The soil is representing the microenvironment, the stroma, the niche that's inside of the tumor that cancer is being exposed to. This slide is reviewing what we discussed earlier, the clonal evolution of cancer. So this is the idea that over time, our cancer itself is going to be changing in its population. And so this is the same idea that we were talking about before, where you have a normal cell, one cell kind of starts to mutate, it kind of rapidly divides, and then we have another cell that mutates and it rapidly divides, and then we have another mutation added and it rapidly divides. So over time, we have all these different subclones within our tumor population. Some of these cells will not be good at invading and doing metastasis, and some will evolve the properties to be able to invade and do metastasis. So we think about this, that this explains a lot of what we see as far as, like if you look at pathology, you see a lot of tumor heterogeneity. If you look at gene expression or even your sequencing of cells, you can see a lot of heterogeneity within the tumors. And therefore, they also have a different ability to respond to different drugs that the tumor is being treated with. And that's why you can see a patient that's treated with a therapy, you see a regression of their tumor, and then, there might be 5 years that go by, and then the tumor comes back up. That's because some of those cells did not respond to the treatment. And so, sometimes just a few of those cells remain, and it takes a little bit of time for that tumor to pop back up. So that's an important concept for cancer and cancer research, is understanding the clonal evolution of cancer, understanding that a tumor is heterogeneous, and how do we treat those heterogeneous tumors. If we have clonal evolution of cancer, that can help it escape surveillance by many different factors that are designed to prevent cancer growth and progression. So our next slide kind of covers that. So thinking about cancer cells and how they remain in the body, we've briefly mentioned a few times that the immune system plays a role in surveying tissue and destroying cells that are abnormal. And so our immune system is keeping our body in check, and it's looking for and detecting cancer cells all throughout our lifetime. So cancer cells tend to present antigens that the immune system can recognize, and then they will target that cell to be degraded. And so if a cancer cell is abnormal, then it typically has abnormal gene expression, meaning in the end that certain proteins or factors are going to be expressed at different amounts, or they could be mutated or altered, and the cell recognizes that. And then and we're going to talk about kind of like what it does just a little bit. We're not going to get too deep into it right now. And what it does is it presents some of that on the surface of the cell so that the immune system can see. And once the immune system sees it, then it targets it for degradation. So one of the key members of the immune system that is doing this job is called the cytotoxic T cell. This is a killer T cell. So this is just one of our immune cells that plays a very big role in immune surveillance and killing of cells that are not normal. So we abbreviate with CTL, and it has the ability to kill an abnormal cell. And that abnormal cell could be abnormal for many different reasons, including it could be infected with the virus, or as we've been discussing, it could be a cancer cell. So we mentioned before that cancer cells have the ability to present antigens. And that term antigen is just referring to pieces of protein. A protein is kind of chopped up, and then you have a piece of it, and the piece of that protein is presented on the surface of the cell. So here is the altered cell, right? And it had experienced a protein that was expressed differentially, and so the cell knows that that's not right. So it chops that protein up and it makes antigens. Then it presents the antigens on a receptor outside of the cell. And that receptor was called MHC. We kind of briefly mentioned that before. So the MHC, the major histocompatibility complex, can present that antigen to the extracellular space. And this is where we have our cytotoxic killer T cells surveying the landscape, looking for any of these antigens to know that, hey, this is a cell that needs to go. And so it has its own receptor called the T cell receptor, and it can recognize these MHC receptors and the antigen. Once it recognizes that, that gives it a clue that this cell needs to die. And so it can activate the apoptotic pathway and cause the cell to die, right? So here we have an experiment that's trying to just determine how important is this antigen presentation process and immune surveillance in the ability of cancer cells to form metastasis. So this is an experiment that was done to kind of do a proof of concept for this idea. So we're going to start talking about this experiment by looking at this middle panel. So here we have a mouse that's injected with A9 cells. So A9 is the name given to these cancer cells. These cancer cells express two surface MHC molecules, H2D and H2K. So it has two of these that it naturally expresses. And so these cells are put into suspension, injected into the tail vein, and then the metastases are observed. So we get a few metastases in this tumor model. We have another model over here. So these are D122 cells, and they express only H2D MHC molecule. They do not express the H2K, so they only express one of them. These cells are put in suspension, injected into the mouse, and the metastasis is observed. Oh, I'm sorry, this was the primary tumor over here. So primary tumor and metastasis. So let's take a look at the difference between these two. So in the first case, we had our A9 cells. They had two MHC molecules. And what happened? They were injected We made the primary tumor, but did we make mets? No, we made no mets or a few mets. It says below, right? Here we have the D122 cells. They only have one MHC molecule. You inject it. We have the primary, and we have mets, right? So what is that telling you? The only difference between these two cells that's being displayed here is that they both have H2D, but this one did not have H2K, right? It didn't have it. So it's suggesting that maybe this was important, right? Because not having it allows for the mets to be made. So they kind of did another experiment just to prove that this H2K would be important for the immune system to detect these cells. So what they do is they take the D122 cells. Remember, they only expressed H2D. They add H2K. When they do that, then that makes it similar to the A9 cells. They inject them into the mouse, and what do they see? They see few mets. So it makes it very similar to the A9 cell model. So this is giving the idea that the presence of the two MHC molecules was important for immune surveillance. They're kind of making the case that with these two MHC molecules, that the immune system can readily detect these cells and kill them, and that is why there's no metastasis. Now, in the case of D122, it did have one MHC molecule, and somehow that was able to kind of evade the immune system, and they were able to go undetected. So this paper kind of made the case that the H2K MHC molecule was very important for the detection by the cytotoxic T lymphocytes. So in our last slide, we were talking about how the tumor cells interact with immune cells. And we mentioned the cytotoxic T lymphocytes. And we were saying that in order for a cancer cell to be able to undergo metastasis, that it is going to have to evade the immune system. Otherwise, it's not going to be able to make metastasis, right? Let's kind of move through this diagram, right? In chapter 2, we even brought this up, right? On this one down here with adhesion molecules. For a cancer cell to be able to do a couple of things. It has to be able to grow in a big tumor mass, or it has to be able to drift off away from the main tumor mass, and that required a change in its adhesion molecules, right? And we had said that if the cell is not adherent to something, then it's going to undergo anoecus. It's going to undergo programmed cell death, right? So at some point, the cancer cell has to evade this process. In order for the cell to move through tissue to begin the invasion process, it has to be able to move through the extracellular matrix. If this is the cell, it has to chop its way through here so that it can move through there. If it's unable to do that, then it's not able to move. And that requires proteolytic degradation. And proteolytic degradation was mediated by the plasmins and the MMPs that we talked about. Those are all proteases, so they have the ability to break down the extracellular matrix. If you remember, the extracellular matrix is made up of things like collagen, and collagen is a protein, so it's going to require a protease to break it up. We're going to have lack of adhesion So the cancer cell is going to have to be able to break away from the primary tumor and be all by itself. That's kind of what we we're just talking about over there. It has to be able to stimulate its own growth. It has to be able to proliferate uncontrollably. So that requires the increase of growth factor expression in those cells. And it also has to have the ability to move. So motility is different from degrading into the extracellular matrix. Motility is actually moving. And so if you remember in cell bio, when we kind of mentioned it in class, in order for a cell to move, then that requires the cytoskeletal components to be there at the leading edge of how the cell is moving, right? And then also in order for the tumor to actually like grow to a bigger size, we said that in order for it to get bigger than 1 to 2 millimeters, that it needed to stimulate its own blood supply. And so that was angiogenesis. And so the cell has the ability to produce proteins that can stimulate angiogenesis. One of these proteins was VEGF, and it was able to stimulate the endothelial cells to proliferate, but also to degrade into the extracellular matrix and to start coming towards that tumor cell. And so that's another thing that the cancer cell has to be able to achieve in order to be successful. For this chapter, we mentioned a couple of things. Our big topics were angiogenesis, invasion, metastasis, evading the immune system. This chapter also just briefly mentions a few more metastasis genes. We covered a few. We kind of talked about cadherin, we talked about VEGF, plasminogen, MMPs. So we've already covered a couple different kinds of genes. There are a whole host of other genes that have been studied that are involved in these processes and that have also been found to be have altered expression in these different tumors. But in general, metastasis promoting genes will code for proteins that stimulate events associated with invasion and metastasis. And an example talked about here is twist. So twist can cause cells to lose adhesive properties and to become motile. Twist is very important for embryonic development. If you think about as embryo is development, especially during gastrulation, we have those cells actually moving around. And so that's a property all by itself, and twist can kind of mediate that process. And so after all of that occurs, and after the embryo has developed, then that twist gene expression is shut down because we don't need those cells to be moving anymore. And sometimes twist is reactivated in cancer and can allow those cells to move throughout the body. Metastasis suppressor genes. So these would be proteins that inhibit events associated with invasion and metastasis. So in this case, we could have a loss of a metastasis suppressor gene, and that can increase the cancer's ability to undergo metastasis. So here we have CAD1 was our example. This codes for an E-cadherin protein. This is a cell-cell adhesion molecule, and loss of E-cadherin can lead to tumor invasion. There's a whole host of other genes that are being studied, but the idea is that if the normal function is to suppress invasion metastasis, if you lose that gene, then that cell has increased ability to invade and metastasize. All right, well that gets us to the end of chapter 3. Please let me know if you have any questions. Thank you.